So this is cell structure and organization for A-level biology for GCSE, for WJEC or EDUCAS. So first of all, you need to know cell structure. All cells are surrounded by a thin membrane made of phospholipids and proteins. Biological membranes are so thin that their structure cannot be distinguished in the light microscope, and in the electron microscope they appear as a single line. Eukaryotic cells contain membrane-bound organelles, which are enclosed areas in the cytoplasm. The advantage is that potentially harmful chemicals such as enzymes are isolated, and molecules with particular functions such as chlorophyll can be concentrated in one area. Membranes provide a large surface area for the attachment of enzymes involved in metabolic processes, and they provide a transport system inside the cell. Oh gosh, I can't stop pausing. So you have ignore my drawing, guys. So, animal cell ultrastructure. You need to remember how to label these. Unfortunately, there's a lot of labels, but that is just the nature of A-level. So you have the plasma or cell membrane. You can't just say membrane. You have to say plasma membrane or cell membrane because there's multiple membranes. The mitochondrion, which is just this little sort of worm shape with a wiggly line in it. Cytoplasm is you can see it's just the empty space. Ribosomes are these little dots. Lysosomes are larger dots. Golgi body is sort of this. It's like a weirdly shaped Wi-Fi symbol. Have a centriole. It's only in animal cells, and it looks sort of like a cylinder. A vesicle, which is small and temporary in animal cells. Smooth endoplasmic reticulum is this structure here. Chromatin are these lines you can see in the nucleus, and this dot is the nucleolus. You have some nuclear pores, a nuclear envelope. It's called an envelope because it's a double membrane, so as long as the lines are parallel, it's a double membrane, so it's an envelope. And then there's rough endoplasmic reticulum with ribosomes. So the ribosomes are on the edges. So it's the same shape with this, but with lots of little dots. And those are the ribosomes attached to the rough endoplasmic reticulum, because it's not smooth. Now, plant cell ultrastructure. You have the nuclear... Let me move my camera back a little bit like that. So you have the nucleolus, the nuclear pores, chromatin, like the same thing, and that all makes up the nucleus. You have a nuclear envelope. Plasmodesmata is only in plant cells, so those are by here. Chloroplasts are also only in plant cells. They're like little squares with lines attaching them in the diagram. Rough endoplasmic reticulum, ribosomes, plasma/cell membranes here. You have a cell wall, you have the cytoplasm, the Golgi body, vesicles, which are these little dots, the tonoplast, which is only in plant cells, the vacuole, which is large and permanent, which are also only in plant cells, and then the smooth endoplasmic reticulum. So when you're looking at images of cells, it's important to understand the units of measurement. The standard unit of measurement is the meter. Biology works in the range of nanometers when considering molecules, to kilometers when considering ecosystems. But for cells and organelles, micrometers are the most convenient unit, and they're like a weird U with an M. So a kilometer, so number per meter is 0.01. So a meter would be 1, a kilometer would be 10 to the power of 3, millimeter would be 10 to the minus 3, micrometer would be 10 to the minus 6, and nanometer would be 10 to the minus 9. So kilometers are used for ecosystems, meters are used for larger organisms. millimeters are used for tissues, micrometers are used for cells and organelles, and nanometers are used for molecules. So 3 principles of cell theory are all living organisms are composed of one or more cells. So one would be a unicellular organism, more cells would be multicellular organisms. The cell is a basic unit of life, and cells can only arise from pre-existing cells. The nucleus is in both animals and plants. It's the most prominent feature in the cell. It's usually spherical, and it contains DNA, which with protein comprises the chromosomes. The chromosomes direct protein synthesis because they are the site of transcription. The DNA also provides a template for DNA replication. The nucleus has a number of components. So it's bounded by 2 membranes called the nuclear envelope. You'd refer to it as a nuclear envelope rather than nuclear membrane because the nucleus is surrounded by 2 membranes. And it has pores which allow the passage of large molecules such as mRNA, which is messenger RNA, which leaves the nucleus through the nuclear pore, and ribosomes. The outer membranes, what is this? They're missing an I, so it confused me. The outer membrane is continuous with the endoplasmic reticulum. First page, yay! So the granular material in the nucleus is the nucleoplasm. It contains chromatin, which is made of coils of DNA bound to protein. During cell division, chromatin condenses into chromosomes. Within the nucleus are one or more small spherical bodies, each called a nucleolus. They are the sites of formation of rRNA, which is ribosomal RNA, I believe. And it's a constituent of ribosomes, so it makes up ribosomes. So you can see here, nuclear envelope. There's a nuclear pore here. It's a bit difficult to see, but this is the gap there. Nucleoplasm containing chromatin, and a nucleolus, and there's one present. Mitochondria are in both animals and plants. They're often cylindrical, and they comprise of 2 membranes separated by a narrow, fluid-filled intermembrane space. The inner membrane is folded inwards to form cristae. So those are these little wiggles. An organic matrix, which is a solution containing many compounds, including lipids and proteins, and small 70S ribosomes, and a small circle of DNA, which enable mitochondria to make some of their own proteins and self-replicate. So here you have the 70S ribosomes. This is the matrix. This is the outer membrane. This is the intermembrane space, so just between the 2. It's just in the membrane. Circular DNA, which allows self-replication, the inner membrane, so the intermembrane space is just between the inner and the outer membrane, and then you have the cristae, or crista for singular. The function of the mitochondria is to produce ATP in aerobic respiration. Some of the reactions occur in the matrix and others on the inner membrane. The cristae provide a large surface area for the attachment of enzymes involved in respiration. Metabolically active cells, such as muscle cells or the liver, need a plentiful supply of ATP. They contain many mitochondria, reflecting the high metabolic activity taking place. I said mitochondria then. Mitochondria. Being cylindrical, mitochondria have a larger surface area of a sphere of the same volume. So in other words, their surface area to volume ratio is larger. Compared with a sphere, being a cylinder reduces the diffusion distance between the edge and the center, making aerobic respiration more efficient. Mitochondria on, if you have any images or diagrams of them, they can appear either longitudinally or like a transverse section, which looks more circular like that. And the reason is because they could either be cut at 2 different angles or planes, because they are a cylinder. If you cut them lengthwise, then they'll look like that. If you cut them this way, they'll look like that, in like an image. And they're also at different stages of development. They can be different shapes, so this is more of an early developed one, this is later. So chloroplasts are only in plants, and they occur in the cells of photosynthetic tissue. Not all plant cells contain chloroplasts. Root cells, for example, being underground, don't need them. In many plants, the highest concentration is in the palisade mesophyll cells, just below the upper surface of the leaf. Each chloroplast is surrounded by two membranes comprising of the chloroplast envelope. So the stroma is fluid-filled and contains some of the products of photosynthesis, including lipid droplets and starch grains, which can take up a large part of the stroma. Like mitochondria, they contain 70S ribosomes and circular DNA, which is here, which enable them to make some of their own proteins and self-replicate. Within the stroma are many closed flattened sacs called thylakoids. A stack of thylakoids is a granum, so that would be that. And each granum comprises between 2 and 100 parallel sacs. The photosynthetic pigments, such as chlorophyll, are found in the thylakoids. This arrangement produces a large surface area, efficient for trapping light energy. So you have the endosymbiotic theory. Endosymbiosis is a theory that describes the origin of chloroplasts and mitochondria as far as back as 1883. So the division of chloroplasts was seen to closely resemble that of free-living cyanobacteria. In the 1920s, the idea that mitochondria were once independent bacteria were also suggested. So mitochondria and chloroplasts have 70S ribosomal circular DNA. Started coughing there for some reason. I keep doing that, I keep coughing today. It's suggested that at least 1.5 times 10 to the power of 9 years ago, some ancient bacteria with very fluid membranes engulfed others and maintained a symbiotic relationship with them. Some of those engulfed were very good at turning glucose and oxygen into ATP, so evolving eventually into mitochondria. Some could turn carbon dioxide and water into glucose, evolving eventually into chloroplasts. In 1867, Lynn Margulis produced a paper titled The Origin of Mitosing Cells, which has sustained the idea of endosymbiosis. Now biologists have plenty of evidence that both chloroplasts and mitochondria have their own origins in free-living cells. So endoplasmic reticulum is in both animals and plants. It's an elaborate system of parallel double membranes forming flattened sacs with interconnected fluid-filled spaces between them, called cristernae. The ER, so endoplasmic reticulum, is connected with the nuclear envelope. This system allows the transport of materials through the cell. So there are two types of ER: rough ER, so RER, rough endoplasmic reticulum. It has ribosomes on the outer surface and transports the proteins made there. Rough endoplasmic reticulum is present in large amounts in cells that make a lot of protein, such as cells making amylase and the salivary glands. Smooth endoplasmic reticulum, SER, comprises membranes that lack ribosomes. It's associated with the synthesis and transport of lipids. Cells that store large quantities of carbohydrates, proteins, and fats, including liver and secretory glands, have extensive endoplasmic reticulum. So you can see in the rough endoplasmic reticulum you have ribosomes attached, flattened